Thanks a lot. First of all, I should kind of probably thank the organizers because I really liked this workshop so far. It was really, I learned so many things. I didn't understand every single thing, but I learned a lot. If you don't know where this is in a small town in the north, and this is how our institute looks like now, when I have left, and this is how it looks like in November, so it's a very warm and cozy place. And I want to speak about kind of evolutionary dynamics in small networks, and this is like in a way, it's the Moran process on graphs, or variants of it. And kind of when Dominik Wodarts gave a talk, which was a very good introduction, so I could delete lots of slides, he said, oh, these are artifacts, that's why I'm not working on this. And I'm talking about these artifacts. I want to understand why these models behave like they do. So we heard a lot about population structure in cancer, for example, in these colonic crypts, and then kind of you can model these and you can ask, why is it this way? Or you can look at spatial structures and ask, how should you sequence something to see kind of all these different colors in here? So population structure is definitely something that is relevant in cancer, but I will not go into these details. So for me, it's more like a motivation to look at population structure. I could have given a cancer talk, but that would have been much less mathematical, I guess. And this is something where I really think there are some open issues that are potentially interesting. So how does tissue structure affect cancer evolution? And this is a super simple model, and there are two versions of it. This one here is kind of from Martin Nowak's group, and there's a arrangement of cells, and then he says, I select a cell proportional to fitness for reproduction, it divides, it shifts the other cells down, and then something falls off. And that's one variant of a process. Another one here is from a kind of a paper from Natalia Komarova, where the process is actually the other way around. So you first remove a cell, and then the neighbors compete for that site. That sounds like a subtle difference, right? It shouldn't really matter in the end, but it matters a lot. And I will later go into this in more detail, and this is often called birth-death process, and that is death-birth. And for the purpose of this talk, and in general I try to do this, I use big letters for selection and small letters for random choices. So this is, I first choose something proportional to fitness to give birth, produce identical offspring, and then I choose kind of a neighbor for death, or in this case, somebody from the right-hand side. And this is, I choose a random individual to die, and then the neighbors compete for that site. So the question is, the argument here was always, this is how tissues are formed. For example, the colonic crypts has the stem cells down here, and then they push these cells out, and they are kind of lost frequently, and that reduces the mutation load. And the question is, should this actually be the case? So should tissue structure actually suppress evolution? And we played around a lot with small networks where we created kind of things, and we asked kind of, are these actually amplifiers or suppressors of selection? I will come to this a bit later. And this is motivated by a paper by Aritz Lieberman about 20 years ago, where he looked at such structures, and he compared the probability that a new mutation fixes in this population to the well-mixed case. And this question, is it actually good to suppress selection, has a lot to do with this distribution.
Because in these models, it's the random choice of which cell it, say, dies, right, or reproduces, is that like proportional? Is it uniform over all this? It's
uniform for this purpose. For now, it's
you don't weight it according to the number of neighbors? Or
no, not yet. Not yet. So what is important in this context would be the distribution of fitness effects. And that's something that evolutionary biologists discuss a lot, and that is, I think, kind of less kind of known in cancer evolution. Kind of what are the mutational effects that you have? And for somebody like me who comes from evolutionary game theory, frequency-dependent selection, you would actually argue maybe that the fitness effect of a mutation is not fixed over time. There's one example where we worked on a cancer where you always see a driver mutation, but you also need certain others, and they're a bit arbitrary. And that would be a case of epistasis, where the fitness, sorry, this is a bit hard to see. So the lower line here is kind of, you have a driver mutation, here you have no driver mutation. And there's something we call secondary driver. And only if you have enough of them, the driver mutation fitness kicks in. And that means for a long time, you just have to accumulate these kind of neutral mutations up here. And then at some point, you can have the driver mutation and fitness kicks in and the population explodes. So that would be a case where the distribution of fitness effects is actually not a concept that makes a lot of sense, because it depends on where you are in this space. So first of all, I want to speak now about fixation probabilities and times in graph-structured populations. Then I will speak about how to construct certain special cases. Then I will go a little bit into long-term evolution and structured meta-populations, which are a bit more complicated from the start. So before I speak about structured populations, I have to speak about unstructured populations. And that's, for example, here I have a mutant, and then I ask, in some evolutionary process, what is the probability that this guy takes over the whole population? So I only have two types. One is the wild type with fitness 1. The other one is a mutant with fitness R. And I put these things on a network, and for an unstructured population, that means that I can connect every cell to every other cell, because everybody can replace everybody else. This is something that when I talk to biologists, they are incredibly confused that this is unstructured because it's such a structure. But it's just everybody is connected to everybody. So one time step in this Moran process would be I start from this population. I select one individual for birth proportionate to fitness. Then I select one other individual for at random for death, like uniform. Everybody can die with the same probability. And then I replace, and then I've changed the state of the population. Now there's one mutant more. So what we then try to do is we put this on these networks, and then we can start simulating, and then we see that the mutant somehow spreads in some way, and in the upper case it eventually takes over the population. And you can actually calculate this. This is a classical example from population genetics where you can calculate this fixation probability, and this is 1 minus 1 over r divided by 1 minus 1 over r to the n. And then some people say, yeah, r is usually larger than 1. It's a mutant that is fitter, and n is typically large, so you can just neglect this guy, and it's 1 minus 1 over r. And then you can, a lot of population geneticists, they prefer not to have r, but 1 plus s. If this would be 1 plus s, then the fixation probability is just s, the selective advantage. And now you can argue for a long time if it should be s divided by 2 or not, or what the corrections are, but depending on how you do these approximations here, this leads to some fixation probability.
So that's a complete graph, is what you're saying?
That's a complete graph, yeah. And of course, then it's a random process. In the lower case, you just lose the mutation at some point. And in this case, you could also calculate this extinction probability. This is, of course, 1 minus the probability that you go to fixation, because you have to end up somewhere. And this is then, in this approximation, approximately 1 over r. So if you have a mutant that is twice as fit as the wild type, it has a 50% probability to take over the population or to go extinct. So the question is, what happens if we have some sort of network? And ideally, I would like to do the same. I mean, of course, I can simulate, but I can only simulate very small networks. And then I would like to know what is the fixation probability. And ideally, I would like to calculate this. And I can calculate this for very special structures, but so far not in general. And again, I can have fixation or extinction. And you see in the spreading process, of course, here, the orange mutant cells, they always form a cluster, because you can always invade only a neighbor, and so you have to invade this network as a cluster. And then you want to calculate this extinction probability. And that's per se, a difficult task. You can do this under certain approximations for certain networks, but in general, that's not easy. So again, this is an unstructured population, the complete graph, and then you calculate this probability as fixation probability. So you plot the fixation probability as a function of mutant fitness. And then there's one here, which is the neutral mutant. So the fitter mutants have a certain probability to take over. For example, for 2, I said it's approximately 50%. For neutral mutants, this is exactly 1 over n. This is, of course, you have to look at the limit here. And if you go to the left, then at some point the fixation probability is 0. And now there are special structures. For example, this one here. And I know that this is not a biologically relevant example, but this is the most extreme case, and I think we can learn a lot from this extreme case. So if you would calculate the fixation probability of this guy, you would find approximately the same thing, but instead of the r, there's an r square, which means that the fixation probability is always in this green area here. It's above the blue line on the right-hand side of neutrality, and on the left-hand side, it's always below. So with this structure, you can increase the probability that a mutant with a certain fitness advantage takes over. And that sounds somehow cool. We always thought that this is something that maybe we can use this in biotechnology or so, but this is far away. First, we want to understand this.
Is there some property of the graph, like the largest eigenvalue of the Laplacian, that is related to this fixation probability?
It's not that simple. So I'm not sure if there is something, but people have tried all kinds of things. And so far, there's no clear rule how you can actually look at this. So what we do is typically we calculate things for certain simple cases, and then we do brute force computation to just look at more graphs and to see what can happen. One other thing that I found very interesting, and this is actually, I never planned to work on this. I found it completely irrelevant, but then somebody came to me and said, look, I have amplifiers of selection and they make things very slow. Something is wrong. And so if you look at this, this is time on a log scale, and this is the number of mutants. So if you have the well-mixed population, I said, this is okay. This is R equal to 2 here. So for a mutant that is twice as fit, you have 50% probability to take over. So if here you have a mutation that goes extinct, you have another mutation at some point, and 5 of them eventually take over. That's my 50%. On this structure here, the ring, I have now 4 out of 10 that make it. And on this, I have 9 out of 10. So this is an amplifier because many of these trajectories end up on top. At the same time, this takes an order, 2 orders of magnitude longer than the blue one, than the well-mixed case. So the price for increasing fixation probability is that you have to wait for a very long time, which is a bit sobering if you think about, say, biotechnology or so. From this, we somehow kind of thought, okay, this is maybe interesting because we can look at kind of selective advantage of a mutant here in a network of size 50, fixation time. So this is now the average time this process takes. I didn't delete these slides because somebody here was interested in time scales of this. So I thought I can talk about it a little bit. So the fixation time here is by far largest for the star graph. Then it's a little bit lower on this ring and even lower on the lattice here and lowest in the well-mixed case. And with this result, I had the ingenious idea. I had a new student. I said, okay, you just proved that you remove a link and that slows down networks. And the PhD candidate, Laura, said, yeah, sure, I can do it. And after 2 days, she came back and said, you're wrong. Because I have a counterexample. And the counterexample is a very small network here, size 4, well-mixed ring, and this we call it diamond. And this one is actually slowest. So sometimes removing a link can make things faster or slower. Which is somehow, for me, it's still, yeah, it would be so much nicer if it would be easier, but it's not. So yeah, this is the slowest, and you can order them here. And the same holds actually you can put these structures on a kind of lattice with periodic boundary conditions. And if you have periodic boundary conditions, this effect is actually gone, I think. And if you have boundaries, it's still there. So it's a bit subtle, and it's a very small effect on these bigger lattices. But you have this, we don't really know what removing a link does.
So is there any specificity in the sense that some links when you remove it improves time and some links when you remove it increases time?
Yeah.
Okay.
We have this observation, but we can't explain it.
But just to understand the process. So you'll pick a random vertex, okay, and you make it into a mutant at random. If it was already a mutant, then you just do it all over again. Just ignore it.
Yeah. So the process here would start in the following: you put a random node is just changed to a mutant, right. And then you do this process. You pick somebody proportional to fitness. That could be this guy. It could also be this guy. And if this is now chosen proportional to fitness, it produces identical offspring that is moving to a neighboring site.
And the neighboring site, and if the neighboring site was already the same color,
then nothing changes.
Nothing changes.
Yeah. So most of the time, nothing changes, especially on large networks. So then we were encouraged to speak about open questions. That is actually an open question. Is the complete graph fastest? So this is a plot of the fixation probability versus the average fixation time for all graphs of size 10. And then you realize you can sort, for example, the star graph that I had is here. And there's something we call a double star. There are things that are having a very small fixation probability. And we would like to know whether it's true that the bottom point of this graph is always the well-mixed population. This is size 10. Then we can do this for different fitness values. This is size 6. This is size 8. It always looks as if this would be the lowest. And we would like to have a statement on this. But so far, we have failed. Where we made a bit more progress, but I think it's still an open question, is an easier question. Does removing a single link from the complete graph always make it slower? We know that this is true for these graphs of size 4, and they're ridiculously small, because the diamond is slower than the well-mixed population. And it's harder then to move on with this, but we definitely know that down here, from numerics we know, that down here the complete graph is the fastest. So we would like to know the complete graph minus one link, is it slower? And this sounds almost trivial, and it's an amazingly hard problem. But I've recently discussed this with Mark Broom from London, and I think he has a very good idea how to approach the problem, but somewhere on my hard disk I have many pages of mathematics that are maybe beyond me.
So I'm maybe missed something. So when you introduce the mutant, does it matter it must matter what node you introduce the mutant at?
We'll come to this later.
Okay.
Very good question, very good question.
Just a simple question. Sometimes the probability is larger, sometimes it's smaller.
Oh, okay, so sorry, I should have mentioned this. This is fitness 0.5. This is a disadvantageous mutant.
Or disadvantageous.
Yes, so that means that the fixation probability is always smaller than neutral, so you are on the left-hand side of this line. So how can we explore larger graphs? And the problem is there are many of them. And this is the number of graphs. I mean, these are unweighted connected graphs of all these sizes. And I've shown you some examples for 10 and for 6 here, where there are already 112 graphs. And this explodes quickly, so even 30 is more than particles in the visible universe. So this is, of course, mathematicians are not surprised, but it's clear we can't simulate it all. What we can do is we can analyze all networks up to size 10. That's a few years ago. Maybe now we can go to 12, but not more. And we can compute fixation probabilities numerically up to n equal to 23 for general graphs. That's ridiculously small. I mean, we can get ideas in this approach, but it's not something that is really satisfying. So another approach that we've taken a few years ago is that we actually use these are not really tools from demography, but this is how I worked with a demographer, and he said, okay, that's how we do things, and let's do the same. And we just said that every node of the graph corresponds to an age graph. So this is how a demographic model typically looks like. There are reproductive adults here in age class 2 and 3, and they produce babies. They produce babies alone. This is just an asexual model. They produce babies at a certain rate, and they age again, and then you have post-reproductive nodes here. And you can actually think about this as this network model. And then you can approximate things, and you can actually do something like diffusion approximation for these graphs. So this is the ring graph, then you have the well-mixed, and you have different updates here. So this is the death-birth updating, death-birth, birth-death, birth-death, and these are, fixation probabilities. These are different sizes. This is n equal to 5, and this is n equal to 50. And the fixation probability in this approximation is this, and it's somehow approximated. So now I use this 1 plus s for r, is s times ne, which is called the effective population size, divided by n. So if you want to write down a stochastic differential equation, sorry, this is a physicist slide. I should have changed that. That's how sloppy physicists are writing down stochastic differential equations. So there's the deterministic term, and there's there's the term in front of the noise here. Sorry about that. Okay. What you can do is now you can calculate the effective size of graphs. And what you see in these examples, we have a couple of special graphs. We have the complete graph, the wheel graph, the ceiling fan, as we call it here. Then we have a k-regular graph, a star graph, and a line. And we can calculate the effective population sizes for all these graphs with this approach. And then you realize, for example, the star graph here, it has twice the effective population size compared to the well-mixed. And it means that it would amplify selection through the reduction of drift.
Wait, what does the effective population size mean?
Good question. Effective population size in this context is a factor that you have here in your fixation probability that you could calculate. I mean, some people say it's the number that you have to choose to make things become correct. But effective population size has many different definitions in biology and population genetics. But it can essentially here it means that we're looking at a different ratio between drift and selection, genetic drift.
So NE is the diffusion coefficient.
Yeah, it's down here.
Yeah. So it's a fluctuation on a stochastic problem. Yeah.
So here you can see the star has a much larger effective population size. If you use birth-death process, you can see they always have the same effective population size except the star. And that means that their fixation probability in this approximation is approximately the same as the well-mixed case. You can take all these networks, they look very similar in fixation probability. And that's a result that reflects a paper by Ben Edlem in 2014, where he said all the fixation probabilities for all arbitrary graphs, they fall into a small area around the well-mixed case. But this is only for birth-death. And you can also kind of look at the death birth here, and you see that kind of for the complete graph, you get something, and here you get effective population size 1. And that's a very small effective population size, and that makes it a very powerful suppressor of selection. So I didn't say that. Suppressors of selection are those that don't go through my greenish areas, but through the red areas. So they are on the right-hand side, they are below the fixation probability of a well-mixed case. On the left-hand side, they're above. So effectively, they reduce selection. So how do you construct amplifiers and suppressors of selection? So before I go into this, I briefly wanted to mention this isothermal theorem, and that's from this paper by Ares Lieberman. So you look at the fixation probability on a well-mixed population, and then they write nicely here, there's an even wider class of graphs whose fixation probability is rho 1, and then they define the temperature of the vertex as the sum of the weights of the incoming links of a graph. So they work in weighted graphs. For us, this is always 1, so you can think about the degree. And the vertex is hot if it's replaced often and cold if it's replaced rarely. Then they say the isothermal theorem states that an evolutionary graph has fixation probability rho 1 if and only if all vertices have the same temperature. That's a powerful theorem. For example, this is an example for this. If you have this circle that we've seen yesterday in your talk, this is something which would have the same fixation probability. What I find a bit disappointing is the way that this is written down, because here in the main text of this paper, they mention the isothermal theorem. In the appendix, they have a theorem which they call circulation theorem, which is it looks more complicated. It's a generalization. And then they just write, the isothermal theorem is just a corollary. And this is kind of, if you write papers like this, they are very hard to read for non-mathematicians. Because essentially, kind of, if you have a theorem, you want to see a proof somewhere, not just say, oh, this is just a trivial result. But this is an important result in this context, because you can see that kind of many graphs would actually behave like the well-mixed case. But if you want to construct amplifiers, and we have the star graph, but in this paper, for example, this was mentioned, this is the so-called superstar, where you have kind of special nodes down here, and then you have these leaves, and the leaves somehow produce into this orange node, and only the orange node can kind of produce into the center node. So these initial amplifiers, they are really kind of special sophisticated structures. I have no idea how to get them, but somehow Ares Lieberman in those days, he had an intuition how to construct things. Okay, this is called funnel, and this is called the meta funnel. So you can have all kinds of weird structures that are amplifiers. And from this kind of, initially I thought, okay, this is maybe not very interesting. But at some point we asked how complicated it is actually to make them, to make amplifiers. So this is now kind of, again, the well-mixed case. And also kind of isothermal means that the ring and the complete graph, they have the same number of incoming links and outgoing links. Okay, the same degree everywhere here. So they have to be isothermal, so they just have the same fixation probability that the blue line here. And then you look at these other graphs. And then you realize, oh, the star is an amplifier of selection. You knew that before. But also the diamond, I have it here, it's also an amplifier of selection. And this one, we call it shovel. Others might call it guitar. So this is also an amplifier. And the line graph, without periodic boundary conditions, is also an amplifier of selection. And I found that strange. I found that strange for two reasons. First of all, I thought amplifiers are hard to construct. And the second thing is, I thought there's much more than amplifiers and suppressors. Because an amplifier is a graph that only goes through the green area. A suppressor goes only through the red area. But why not have graphs that have intersections here? And what seems to be logic to me is the fixation probability has to increase in the fitness of the mutant. But still you could have intersections, right? You could go above here, and then like this. But we don't find them, at least not for this size. So then we said, okay, we have to do a bit more. We have to work on other graphs. I said, okay, let's look at Erdős-Renyi graphs. And Erdős-Renyi size 4 is, yes, okay, that makes no sense. We have here the probability of making connections. So everything that is white is unconnected. And size 4, I mean, these are just the 6 graphs I just showed. They have kind of green is kind of amplifiers, and blue is isothermal. So if everything is connected, everything is blue. In between it's green, but we don't see any other colors because we only have 6 graphs.
What do you mean by probability of link connection? Is that how you construct them?
Yeah. So the Erdős-Renyi graph is a certain probability that you have a link. Here you see a couple of red ones that are suppressors, and yellow ones, we couldn't classify them. We just said these are other graphs. And in larger graphs, you see even more of them. But if you switch to death-birth, so birth-death is the one that I had earlier. Sorry, I should have switched this maybe on the right-hand side, from this Novak paper, and the death birth is, I explained this with Natalia's paper. Now everything is red here. Now everything is a suppressor of selection. So depending on this process, you can have very different dynamics. And in this case, actually, we found only suppressors or disconnected graphs, suppressors, and some isothermal, and only this yellow class up here, which is also here, but it's very small. And this is actually a graph that is always reducing fixation. I come to this a bit later. And since this is an old paper, nobody wants to read it anymore, but you have to read the title here. So most undirected random graphs are amplifiers of selection for birth-death dynamics, but suppressors of selection for death-birth dynamics. I found this interesting that with this change, you can immediately change this class entirely.
Although it's the same for complete graphs, right? In either case?
Yes. Complete graphs and isothermal still holds, sorry, no, isothermal theorem holds here. It holds only on the left-hand side. The isothermal theorem is not something that you can immediately transfer to the death-birth process.
P equals 1 gives you a complete graph.
Yeah.
Very
yeah. Yeah. So if you want to categorize all networks of size 10, you find something similar. So initially we worked with random graphs, and at some point we had enough not computation power, that was always the same, but we had enough mental power to program something that could actually do this. And that was done by Marius Möller, who was a master student, and he constructed all of them and looked at this classification, and essentially find the same. Birth, death, everything is green, everything is an amplifier. Death, birth, everything is red and a suppressor. So yeah, if you have this spatial Moran process, you have to make these choices. You can have, of course, birth, death, and death, birth, but you could also have selection for death or at death. You could have choose, for example, for the inverse here. You could choose at both birth and death. You could also do it randomly. Then you have to ask, what do I do first? Do I first choose somebody for birth from the population and then enable to die, or do I do it the other way around? And again, you can have selection in all these steps. Then the question that you asked earlier, where do we put the initial mutant? I mean, here it doesn't matter, but here it does matter where we put the mutant.
Well, you could average overall possibilities.
Yeah, that's what people normally do. You put uniform probability, and then you average over the fixation probabilities. And everything I said so far is working with this. So if I set the fixation probability on some random graph, then it's averaged over all positions. You could also ask, during a mutation event, where do you put the mutated cell? Because normally you have I speak about cells here, but you have an individual that has a mutation, and then normally the assumption is that the mutation is the individual that you put somewhere else. But you could also say, no, the mutation stays with the mother cell, and the unmutated cell is moving. And that makes a difference. So the problem is this all leads to a single well-mixed Moran process. So it's very well defined here, and here you have a zoo of models with lots of choices. So the only thing I really want you to remember is it makes a hell of a difference how you simulate space. So space is not just something that we turn on and off. In this kind of models, it's just complicated what you do. So amplification or suppression of selection is not a graph property, but it really depends on structure and dynamics. And many of these subtle differences really matter, and the kind of models we are working with, they are not robust to these changes in detail. So this is why Dominic is wiser and chooses to work on the contact process. But if you want to have more robust models, for example, you could say that all these nodes are small subpopulations and you allow fluctuations in size, and then you can do much more. And that's something that Anne-Florence Bitbol in Lausanne is doing very successful. She's introducing these models and working with them. And I know they are more robust, but still I want to know why do so many things go wrong? So and now I'm maybe moving away from biology more and more, but at some point I really want to find out what these models are doing. Sorry. Now again, I look at Erdős-Renyi graphs, and this is the birth-death process. This is the death-birth process. Sorry, I switched the colors here. AOS, so this is still an amplifier of selection, and this is still a suppressor of selection. And now you could instead say, I put the mutations where there's most change. Because if I assume that reproduction comes with a mutation, then those nodes that change a lot, they're much more likely to harbor mutations. So if I do temperature investigation in Erdős-Renyi graphs here, I find something here which we call it suppressor of fixation. And that means if I compare to the fixation probability of the well-mixed case, it's always below. So it's hard to reach any fixation on these graphs. And on this side here, I have piecewise amplifiers of fixation. These are things that actually, even in neutrality, can lift fixation probability above the neutral case. And this is just by changing the, sorry, I forgot to mention here. This is, this is DBP. So I have a death birth process, and I move the parent. So this really depends on the, shitty notation, sorry. This is I move the parent and not the offspring. So the parent is the one that doesn't have the mutation.
Does it matter how much it suppresses selection? So you have a bunch of graphs.
Yeah, okay. So because of this result of Ben Adlam, most of the time you're close to the well-mixed case.
It's a very small effect.
It's a small effect. For most of these.
Sometimes it's a large effect.
So it depends. You can construct graphs where it's a large effect, but to have a really good amplifier, you have to have directed graphs. So usually it's not a big effect. So, but you can of course move on and say, okay, let's make some natural assumptions. And one is you could just say cells divide according to fitness and their offspring displaces other cells. So you could say, just choose to work on birth-death updating. I know that Natalia assumes that death-birth is more natural, which is also...
do both.
But yeah, but whenever we cite somebody for saying, okay, we could also work on death-birth, we cite you. And then we assume that errors occur during replication, so we do this temperature initialization. And then we can say the offspring cell may include replication errors, so the mutant cell moves. So that's what most people do. So if you have cells dividing, then normally you can't say who's parent and who's offspring. Then that somehow motivates me to say, okay, it could also be the other type that moves. So then let's look at long-term evolution, because so far I only spoke about fixation probabilities. But if you have fixation event, and then you have the next mutation, and you have the next fixation event, then you can ask which structures are actually best in increasing the fitness in the long run. And that's not the same as fixing the best mutations. So in the long run, new mutations will always arise somewhere. And these mutations will predominantly arise in those sites where there's a lot of turnover. So in the star graph, for example, in the center. So if you look at fixation probabilities here for uniform initialization, this is the well-mixed case, this is the star. So then you see that for uniform things, the star is above the blue one here, above the complete. It's here, it's below. So this is what we call an amplifier of selection. Then we can also have a star with self-loops, which is slightly different, but it crosses here. So that's a structure that we would have classified as other earlier. But it's it's interesting here on that side. But if you instead do temperature initialization, if you do this instead, then the star graph suddenly is always below. So suddenly now the star is very bad at fixing mutations. And that is because all the mutants tend to rise in the center, and the center is always replaced if you select a leaf node. So basically, if you have a mutation arising in the center, it's almost immediately dead.
Well, didn't you say that you don't see crossing? Isn't this an example?
Yeah. Initially, we didn't see it, but in this case, now we see it. With the
it's because of the structure of the green.
Because of the structure.
Which is strange. I cannot really see.
Okay, sorry. This is too small. This is a star graph where this is a bidirectional link, but you can also replace yourself in the leaf nodes.
Maybe I missed something, but if you introduce a mutation at the vertex that has the most edges, is that the one that spreads the fastest?
That's first of all, it's the one that is replaced most often, because if the others reproduce, they always want to replace you. So that's if I have time, I could show a simulation. You can play around with this, but you don't want to have a mutation that arises somewhere where there's a lot of traffic of new mutations or new offspring.
About neutral mutants, what is the fixation probability of neutral mutants?
Of neutral mutants, it's on the star graph. On the complete graph, let's start here. It's 1 over n. It's 1 over n for all these 3 cases here. If you do the temperature initialization, it's 1 over n for the complete, because that's just the same thing, but it's very small for the star graph.
So how do you interpret this?
I interpret this as if you would have the structure as a star or a scale-free network, then even neutral mutants would make it with a smaller probability than neutral and well-mixed. So the concept of neutrality in structured populations is problematic. And this is one of the things where I start thinking that potentially what we do here could make population geneticists think. I don't say that this is a good model for anything, but we have certain results that could have an influence on how we think in population genetics. So if you now look at a process where you have kind of a fitness distribution that you just fix, and then kind of a new mutation is just chosen from this distribution, then you can look at a process where you look at the number of mutations here, and then the fitness of the population, and this tends to go up. So we have a couple of trajectories here, but the average is shown here, and then you realize that for the amplifier here, so first of all, I have to tell you what the amplifier is, and for that you have to go back here, because this is now an amplifier at least around neutrality. This is now the green structure, no longer the red structure. The red structure reduces fixation probabilities everywhere. So if you go here, the amplifier initially is very good. Fitness increases fast and stays high, but at some point, the red structure, which is actually reducing fixation probabilities, is taking over, at least reaching this
Please
point.
repeat again, what is temperature initialization?
So in the star graph, you mostly have mutations here.
Well, what is the procedure?
The procedure is kind of, the next mutation arises when, mutations arise during reproduction. That means kind of whenever you reproduce, you could have a mutation. And that means that you mostly have mutations in the center. So in the long run, for these structures, it's actually not important to fixate the good mutations. It's more important to get rid of the bad ones. And that is something that the star graph in this updating is just much better at. So amplifiers of selection, they do not necessarily maximize fitness, which is also somehow casting doubt on my biotechnology idea. So probably that's bullshit. So
what's fitness here, by the way? I'm confused.
Okay, good.
What's the relation between fitness and the probability of mutation? I mean, just all the parameters in here.
So, yeah, okay. So what you do here is kind of you have a certain fitness. And then you kind of, let's say kind of the whole population has this fitness F. And then you run your process, and at some point you have a mutation. And that mutation is actually on the star graph most likely to arise in the middle. And here it can arise everywhere with the same probability. And for this new mutation, you just choose a random number from this distribution again. And then you ask kind of, will it reach fixation or not? And then eventually your population fitness might be a different F. And then you go on with this process. And this is kind of the F is shown here. So eventually kind of you go higher and higher, and you ask kind of, what's the different characteristics of the different graphs? Okay, I already said that the decision where to put your offspring, that can be really crucial. And in this case, for example, if you change from parent and offspring placement who was moving, you change the predominant site of mutations from the center to the outside thing here. So if you have the complete graph in blue here, the green one is the birth-death process on the star where you put the parent, or where the parent moves. And the parent is the one that doesn't have a mutation. That means mostly the mutations arise out here. And in the other one, in this purple thing here, they mostly arise in the middle. That's the process that I explained earlier. So in the long run, these fixation probabilities may not really reflect the steady state. You really have to look at different properties of the graph. I still have very few minutes, right? 2. Okay, then let me jump over this. Yeah, I also jump over this. And then let me briefly say something about structured metapopulations. And what people do for applications, they want to do, in the biotechnology, for example, you have different populations, say microbes somewhere. Or you have kind of patches in ecology. And then you can look at these different structures and ask kind of what goes on here. And there are even experiments now with microbes. And the question is, do we actually allow the population to fluctuate? And if we do this, yes, we can work with these spitball type models. If we don't, we have some sort of evolutionary graph theory of the second kind, where you have you can couple migration to reproduction, and then you have to say, how do I select a patch where something happens? How do I select an individual from the patch? How do I select for death from which patch? And how do I select within that patch? That means you have a whole table of different things, and most of these things are completely unexplored. So people concentrate on very few, and that's where we have references here on this table. So it's a big table of a zoo of models, and most of them are completely unexplored, and I don't think that it's justified to just not do this at all. But what is interesting, that this isothermal theorem can still hold for something. And if you have a structure like this, where you have two patches and they are connected by migration, you could think about this as something where you have two kinds of links, the strong links within the patch and the weak links between the patches. And that means the isothermal theorem is still holding if the patches are of equal size. So if you have a regular metapopulation, everything still holds. So what we're currently doing is we're trying to figure out what happens if we have some asymmetry in these models. For example, if you have different population sizes. If you have two patches of different population sizes, this effectively introduces some asymmetry in the way things work. Okay. What I find a bit concerning is if you talk to people who are doing this all the time, say population geneticists, they often use these packages, and this is an example from SLIM. And the SLIM package just has an instruction, how do you put population structure? And this is like 4 lines. This is how you put population structure into your model. And actually, I'm from this description, I cannot read what they're really doing. But they're doing something, they're making some choice here, and it would be good to figure out what exactly is going on, because these are the people who tell us what is realistic in biology. Okay, so this brings me to my summary. So this evolutionary dynamics on networks has surprising and sometimes counterintuitive properties. And it's easy to construct amplifiers of selection or suppressors, but they really depend on details. Fixation is not the same as long-term evolution, of course, and then kind of moving to metapopulations leads to other new effects. So I'm kind of, at some point, kind of in our department, we started this habit of taking a picture and putting it to the wall for the next generation. So everybody is still here. So there's the old picture here. There's the newest picture, so everybody is still around. And I'm very happy that I work with many brilliant people who can do these things.